There you go.
Sure. Uh, Hi, thanks for the introduction and thanks for the opportunity to speak here tonight. I'm going to talk a bit about the work that I'm involved with to develop cryopreservation and biobanking tools to secure biodiversity and to support reef resilience and restoration efforts across a range of scales. So coral reefs are one of the most important ecosystems on the planet. They actually exceed rainforests in species diversity, and they provide habitat for around a quarter of all marine species. They also form natural storm barriers that help to protect our coastlines, and they support millions of people around the world by providing food and employment. And in doing so, they contribute billions of dollars to the global economy every year. But coral reefs are threatened globally by a range of factors, mostly related to human activities, the most significant of which is ocean warming caused by climate change, which can cause coral bleaching. So most of the color of a coral actually comes from symbiotic algae that live within the coral tissues. And like most plants, these algae photosynthesize and produce things like sugars that the coral host can then use as an energy source. And many corals actually get up to 90% of their energy requirements from those photosynthetic products that the algae produce. But those symbionts are actually quite sensitive. So when water temperature is elevated for prolonged periods, and that can be as little as 1 degree above normal temperature for a period of 4 weeks. So if that happens, they can become stressed, and then the relationship between the symbiont and the coral host breaks down, and the symbionts are ejected from the coral tissues. So as a result, the coral becomes pale because the tissue itself is actually quite translucent. So you can see the white coral skeleton through the tissue. But it also loses its primary source of energy. And so if the coral can't recover its symbionts, then it will die. And so when this happens on multiple colonies in a reef or across a larger region, then we get these mass bleaching events, and they can result in the death of entire reefs. And so the problem there, aside from the loss of those corals, is that the dead coral skeletons can become overgrown with algae and eventually turn into rubble. And so you get this phase shift. And when the reef is destroyed like that, you lose the habitat that supports all of those other fish, all of those other species, sorry, the fish and invertebrates and things like that live in the reef. But also when you lose coral colonies on the reef, you also lose genes from the population. And so that leads to an overall reduction of the genetic diversity out there on the reef, and that can impact on the ability of corals to adapt to the changing environment because they need a lot of genetic diversity to maximize their ability to adapt. And so last summer in Australia was actually the 6th mass bleaching event since 2016, and only the second time that back-to-back bleaching events have been recorded. The first was in 2016, 2017. And you might also have heard about the major bleaching event that occurred over in Igloo Reef in Western Australia earlier this year as well. So this is not something that's just restricted to the Great Barrier Reef. This is something that we're seeing globally. And what we're seeing is that the rate of ocean warming is actually exceeding the ability of corals to adapt. And so the modeling that's being done here in Australia and also by various groups internationally is suggesting that we're likely to see an almost complete loss of coral ecosystems globally by around the end of the century, which is a pretty dire situation. So it's really important that we're acting now to do what we can to help the reefs that are out there and to secure as much of the current genetic diversity as we possibly can. So the most effective way to secure genetics long term is using cryopreservation and biobanking. And so coral cryopreservation in Australia began back in 2011 as a collaboration between Taronga and the Smithsonian Institution in the United States, with the objective of just starting to protect and bank some of the genetic diversity of the Great Barrier Reef. And so there are currently over 4,000 samples from 35 species of Great Barrier Reef corals that are stored in the Taronga Cryopreservation Bank. And these samples have come from corals across 10 different traditional owner sea countries. And so now they're cared for off country in the Cryopreservation Bank, which is in Sydney on Camaragal country. And then there's a second site out in Dubbo on Wiradjuri country. And so we're building on that history of biobanking as part of the Reef Restoration and Adaptation Program, or RRAP, which was initiated in 2020 as part of the federal government's Reef Trust Partnership. And so the goal of RRAP broadly is to develop the technologies and pathways that will be needed to support a future reef restoration industry in Australia. And so it's important to note that all the activities that are undertaken as part of RRAP are done so with the free, prior and informed consent of the traditional custodians of the sea countries where that work takes place. And so I'll talk a little bit more about that later on. But we also collaborate internationally with groups in the United States, as well as some folks in Mexico and in Curacao, on the development of cryopreservation technologies and on biorepository development. So in the context of reef restoration, cryopreservation and biobanking are enabling technologies. So benefits things like greater control over reproduction and genetic management, for example, being able to breed between colonies that are spawning on different nights or in different years, to help support things like selective breeding to develop thermally tolerant corals. And of course, cryopreservation can provide an insurance policy by securing the genetics and diversity, both in research populations, but then of course on those populations out on the reef to prevent extinction. But it's really important that those cryopreservation technologies and pathways are standardized so that those outcomes are reliable, and then they can, and that also that they can be used across a range of scale, from individual users who are cryopreserving a few samples in a laboratory through to like commercial scale processes like what you would see in like the dairy industry or something like that. And then it's also really crucial that these technologies are practical and that they can be integrated into the workflows and pathways that are part of the restoration process. And so when we look at coral, they have a pretty complex life cycle. So there are several stages that we can target for cryopreservation. We're working across all of these sample types with a range of different cooling and warming technologies. But the most mature technology is cryopreservation of coral sperm, which has been possible since 2006 and has been applied in over 50 species of reef building corals around the world. And so that's the approach that's the most translatable, I guess, at this stage in terms of being ready for application into some of the large scale restoration programs. But we can also target coral larvae as well as adult coral tissues or fragments of coral, and of course the algal symbionts. So we've got colleagues in the US at the Smithsonian, at UC Berkeley, at Texas A&M, who are working on cryopreserving fragments of adult coral. But I'll just be talking about the cryopreservation of sperm and larvae and symbionts here tonight, because those are the most important sample types here in Australia and the types that we're working with predominantly. So it's also important to note that coral have some of the most restricted reproductive strategies of any animal species. And so spawning usually only occurs around the full moon in late spring and early summer. So there are only a few nights each year when we can actually access sperm or produce larvae for cryopreservation or for restoration. And so looking at the sperm cryopreservation protocol, the basic protocol that we've used across all of those species is essentially the same. Most of the reef building species that we work with are hermaphrodites, and so they produce, most of them produce an egg sperm bundle. So there's a packet of sperm surrounded by eggs. So those are spawned into the water and then they break apart to release the sperm and eggs. And so we can, on spawning nights, we can collect those gamete bundles and allow them to break up and isolate the sperm. Then we use computer-assisted sperm analysis to look at the concentration and the motility of the sperm in that sample, just as a representation of the quality of that sample. We then combine the sperm sample with a cryoprotectant called DMSO, which helps to pull water out of the cells, and in doing so, it prevents ice from forming inside the cells, because that could kill them. And then we control how we cool those samples. We cool them at 20 degrees Celsius per minute, and we do that using a 3D printed cooling device. So it's actually a nice low tech, low cost system that's really easy to transfer. There's around 5 or 6 groups around the world who are currently using this system, which is nice. And then at the end of the coral spawning period, which is usually about a week, we then transfer those samples to the Taronga Cryopreservation Bank for the long-term storage. So since 2011, since the program began, there have been samples collected from 35 different species from across the Great Barrier Reef. And so the samples that are stored at the Cryopreservation Bank make it the largest collection of cryopreserved coral samples in the world. But most of those samples have actually been collected during coral spawning at the Australian Institute of Marine Science in Townsville. So a lot of the samples we've collected have come from that central region of the Great Barrier Reef. Most of the reef is actually sort of 10 plus kilometers offshore, so accessing the samples or the coral colonies is quite challenging and logistically difficult. So it makes sense that a lot of the material has come from that central region. But we've also collected from down in the southern region around the Keppel Islands and Heron Island, and then up through the north of the Great Barrier Reef as well. So it's also important to note here that the Great Barrier Reef extends across around 70 different traditional custodian sea countries. The samples we've collected have come from 10 of those traditional owner groups. And so we've been working with one of those groups to develop a culturally safe approach to biobanking that I'll speak more about later. So the coral sperm cryopreservation technology has been around for a while, and it's been used to produce coral larvae, but generally that's been on a fairly small scale, and the focus has been on actually collecting those samples and getting them into the biorepository. So less focus on how you then go on and use those samples to produce larvae. So it's really been done mostly at a small scale. And so what we've been thinking about over the last few years is then how do we actually scale up the sperm cryopreservation process, and how much do we need to scale it up by? And so we started out when thinking about this to try and work out what at scale actually means for coral sperm cryopreservation. You know, how much sperm do we actually need to have frozen down to produce larvae at a restoration relevant scale? And so before we started increasing the number of samples we bank or the volume of the samples, we wanted to know what the fertilizing capacity of our current sample unit was. So normally we would collect 1 milliliter of sperm, and that's at a concentration of 10 to the 9 or 1 billion cells per mil in those cryovials. And so we wanted to know how many larvae we could actually produce using that one vial. So normally when fertilization is done with coral, you use eggs at around 10 eggs per mil. So that would be 10,000 eggs in a liter, for example. But we wanted to see what would happen if we increased to 100 eggs per mil or even 1,000 eggs per mil. So getting up to around 1 million eggs in a liter of water. And so that would enable us to keep the volume quite low, and therefore the sperm requirements relatively low as well. And so what we found was that if you have good post-thaw motility and you match that motile concentration of the cryosperm to that of the fresh sperm, then you can actually get comparable fertilization rates, regardless of whether you're using fresh or cryopreserved sperm. We also found that the larvae produced using those cryopreserved sperm had comparable settlement and survival rates compared to larvae that were produced using fresh sperm. So that's a really positive outcome, because it means that firstly, all of those historic samples that we've got that have been banked in that way, they can potentially be used to produce larvae at a restoration relevant scale, but it also means that we don't need to completely reinvent the wheel when it comes to increasing production and upscaling our use of sperm cryopreservation. And so we also currently have some recruits, coral recruits that were produced using cryopreserved sperm using this fertilization approach that have been deployed out onto the reef as part of a large field trial. You can see in the image down on the bottom, these are some young coral recruits after about 3 months out on the reef. They're just inside those black circles. They're quite small. And so this is really a big first step in starting to understand how cryopreserved sperm might be able to be used to support restoration. Okay, so moving on from the sperm, the next life stage that we target for cryopreservation is larvae. And it's actually a lot more complicated than freezing coral sperm because they're a complex organism rather than just a suspension of cells. Plus, the tissues are quite sensitive to cold and low temperatures. So, but being able to cryopreserve coral larvae is potentially a really important step for restoration because it would enable us to store larvae that were produced during spawning and then use them for restoration throughout the year, rather than only having larvae available for restoration during the spawning season, which is what the case is now. And so we started out in this process by using a technology called laser nanowarming. And so we use this kind of as a proof of concept, and it was the first time that coral larvae had actually successfully been cryopreserved and recovered. And what this basically involves is that we put the larvae into a high concentration cryoprotectant solution that contains gold nanoparticles. The sample is then cooled extremely rapidly by plunging it directly into liquid nitrogen, and that causes it to vitrify. So instead of forming ice crystals, it just forms a glassy solid with no ice crystals formed at all. We then hit that sample with that vitrified sample with an infrared laser pulse, and the gold nanoparticles absorb the infrared energy and release it as heat, and so we can warm it millions of degrees per minute. And so that allowed us to recover larvae at a rate of about 50%, which was pretty good. And that was working quite well in some of the species that produce smaller larvae, like your mushroom corals and things like that. And those larvae were able to function normally. They were able to settle and take on symbionts and metamorphose and all of that, which was good. But the main issue there was the scalability, because this is quite an intensive process where you're pipetting larvae around and things like that. So not something that really translates to the large scale restoration that we're trying to aim for. So what we're using now for the last couple of years is working with a technology called Cryomesh. That's been developed by our collaborators at the University of Minnesota. And it's been developed originally with a view to cryopreserving pancreatic islet cells as a treatment for diabetes. And so it's great being able to access some of those technologies, and we're working on being able to translate that for coral. So again, we know that this technology works with some of those smaller larvae, but there are physical limitations to the way heat moves through tissue. So when you get to species that produce larger larvae, like the acroporids, which are one of the main reef building species, then it becomes more challenging. And so we're working through that now and working through those species differences in how the larvae respond to cryopreservation. So we're making a lot of progress in understanding the system, but there's still a lot more work to do to get to the point where we can actually be banking thousands or even millions of larvae. So then the final sample type that we work with is the coral symbionts. And so these cells are actually quite challenging to cryopreserve. They have a lot of internal complexity mostly because of all of that photosynthetic apparatus that they use. But they also have a really tough outer membrane that acts as a barrier to the movement of water and cryoprotectants. So it's really hard to prevent that ice from forming inside them. And so we've been working with the symbiont culture facility at the Australian Institute of Marine Science to start biobanking down the cultured symbiont strains that they have developed there. So As I mentioned earlier, the algal symbionts are quite sensitive to temperature, and so there's been a lot of research to develop symbiont strains that can survive at higher temperatures. And so this process is called assisted evolution. It can take hundreds of generations over many years to develop these heat-adapted strains. And so cryopreserving these strains is an important insurance policy in case an incubator fails, for example. But it also means that you don't have to maintain every single strain in living culture. You could instead bank some down and recover them when you need them. And that's essentially how stock centers are managed for research animals like zebrafish and mice and things like that. And so using a slow cooling protocol with cryostraws, so similar to what we do for coral sperm, we've been able to successfully cryopreserve and recover quite a broad range of symbiont cultures across all of the major Symbiodiniaceae genera that are associated with coral, and that includes some of those heat-adapted strains. And so the goal now is really to optimize that protocol and standardize the workflows to create a biorepository that can support that thermal adaptation work. Okay, so just shifting now away from the technology aspect to a bit more of a bigger picture view of how cryopreservation and biobanking are used in practice. And in particular, the cross-cultural approach to biobanking that we've been developing. And that really sits across all of the work that we do. And so this work began in November 2022 when we were fortunate to participate in an on-country spawning event, which was at Konomi or North Keppel Island in the southern Great Barrier Reef. And that was organised by scientists at AIMS as part of the Australian Coral Reef Resilience Initiative and as part of the Woppaburra Coral Project. The Woppaburra people are the traditional custodians of that Keppel Islands region. And so we were able to set up a mobile laboratory in the classroom and the Konomi Environmental Education Centre. So you can see that in that top picture where we're set up on some kids' tables and chairs, which is the usual groups that they get through there. And so that allowed us to cryopreserve the coral samples and work through that process. And then it also allowed us to demonstrate the cryopreservation process, and it was a great way to provide first-hand experience of what was involved, and allowed us to have some informal discussions around the cultural concerns that Woppaburra people had around the biobanking and the transfer of samples to the Taronga Cryodiversity Bank at the end of spawning. So it's important to note that a lot of the traditional custodians view all things as being part of country. So living and non-living, tangible, intangible, if it comes from country, it is country and it remains a part of country regardless of where it is located. So what we're essentially doing when we bank these samples and we store them at Taronga is that we are removing material from country and storing it on the country of another group. So these informal discussions that we were able to have on Konomi led to the conception and initial planning of a biobanking cultural safety event to enable members of the Woppaburra groups to visit the Taronga Cryodiversity Bank and to continue those discussions around cultural matters. And so in September 2023, we held a biobanking cultural safety event at Taronga, and that included Woppaburra traditional custodians, members of the Australian Institute of Marine Science Indigenous Partnerships team, some of the Gamay Rangers from here in Sydney, who are a local sea ranger group who care for the Botany Bay or Gamay, and also from the Taronga cultural programs and science teams. And so the main points of discussion were really around how we ensure that the samples that come to Taronga maintain their cultural links to country. And so one of the ways that was achieved was through the creation of a cultural piece that the Woppaburra representatives put together. And so that was produced by putting sand from Konomi Beach into these jars, along with gum leaves that were used in the Welcome to Country ceremony that was held for the event. And they were mounted in a piece of driftwood that had come from Konomi as well. And so you can see in that image on the top right there, the Wapabara traditional custodian representatives holding that cultural piece at Taronga. And so that cultural piece stays at Taronga and accompanies those Wapabara samples. And so some of the key outcomes that came from that were around really that the Gamay Rangers will be the cultural custodians of those samples while they're at Taronga as representatives, local representatives of the marine environment, but that also that country and contact information will be maintained in the sample databases and linked to those samples so that we always know where those samples came from and that we can refer back to those groups for permission in future use. We also have provisions in the materials transfer agreement to ensure that anybody who's requesting access to these samples must have free, prior and informed consent from the traditional custodians where those samples originated from. But then also that there will be regular review of the information and the permissions to hold those materials at Taronga. And that includes the opportunity to withdraw consent and have those samples returned to country if requested. And so these outcomes and discussion points provide kind of a starting point for a framework for culturally safe biobanking practices across Great Barrier Reef sea countries more broadly. And so we're hoping to take this approach to some of those other groups and have these similar discussions with them. And so that'll be a really big part of our work going forward as well. And then looking beyond Australia, there is more and more interest from people around the world to start biobanking on their local reefs as those impacts on reefs around the world become more and more apparent. People are looking for solutions and options, and biobanking is something that a lot more people are starting to think about now. And so it's really important that the technologies that we're developing are really transferable, that they can be taken by other groups and implemented and used reliably. So we're doing that in a couple of ways. So we're part of a grant funded by the Coral Research and Development Accelerator Platform, which is an initiative of the G20 group of nations. And that's with partners in the US, Mexico, and Curacao. And part of that was to help the folks in Mexico and Curacao to set up biorepository infrastructure that they need to act as regional nodes for biobanking in their areas. But it's also to develop the training tools that are needed to support new users. So if people want to learn how to biobank, there are resources available for them to learn. And so we're currently working on a training module for the sperm cryopreservation process. And there will also be a second module for the larval cryopreservation once we've got a reliable protocol for that. So we'll be running a workshop in Mexico in October this year with attendees from the Dominican Republic and Colombia to train them in sperm cryopreservation so that they can start biobanking. And then these training tools will also be useful in Australia here. So next year, we're planning to run a biobanking workshop during spawning at Ningaloo Reef over in WA. And so we want to be able to develop some local capacity over there. And so those training tools will be really useful for that. There's also a couple of international working groups that I'm involved with, the Coral Biorepository Alliance, which is more focused on biorepositories of living corals in aquaria, and also the Coral Restoration Consortium Biorepositories Working Group. And so these groups are really good ways to communicate with the coral restoration community so that they're and to reach out to those groups and to make those resources available to people who want to start biobanking and to understand what is involved in actually setting up a biorepository. And then as part of the CORDAP grant, we're also working with the United States Department of Agriculture, National Animal Germplasm Program to adapt their animal grid database system for use with coral. So it's built specifically for managing cryopreserved samples. So we've got a data scientist here at Taronga who's helping us to get that database online, and then that will be a resource that's available to groups around the world to be able to manage their samples, because the samples that you have in your biorepository are only useful if you know exactly what they are and where they came from. So the database is a really crucial part of that. Okay, so I think that's pretty rapid overview of all the different things that we're working on and things that are happening across the coral cryopreservation and biobanking world. So there's a few links here. There's a YouTube link that shows some of the on-country spawning work and talks about the Wapabara Coral Project a bit more. And then there's some other links both to the RRAP program and then also to the Taronga Cryodiversity Bank. So yeah, thank you for your time. I'm happy to answer any questions and we'll have a bit of a chat.
Yeah, thanks so much, John. We'll launch straight into the discussion, and I think the discussion will follow kind of very much the processes of the slides that you've talked us through as well. So I've been introduced at the top, and John's given a description of a great overview of the Reef Restoration and Adaptation Program, which supports this amazing program of research. And just recapping that the objective of the program is to provide decision makers, be they reef managers or reef community, traditional owners, with options for intervening at scale on coral reefs to help them avoid, recover from, and adapt to the effects of climate change. And of course, the toolkit needs to work alongside climate mitigation and best practice reef management. But I think, John, the scale is one of the key words here. And we're in a bit of a race against time, as we know. There's been a lot of media at the moment that everyone will be aware of. And we're looking to support with the challenges the reefs around the world, not just the Great Barrier Reef, are facing. And we're looking to support a truly enormous and complex ecosystem. So the work that you've described is coming from one team working out of one facility.
Yeah,
you sort of touched on it, but what's the long-term vision here? When you say a restoration relevant scale, what does that mean? How big can we go?
Yeah. I mean, yeah, it's really quite a daunting task to think about kind of the scale of what we need to do. The Great Barrier Reef is 2,000 kilometers long, 2,000 or more, 3,000 individual reefs. And so it's really difficult to kind of work across that scale. And so everything that we do has to be able to suit that kind of a process. So I mean, in terms of the cryopreservation, it's really about working with those groups who are developing the coral aquaculture, who are developing the technologies to deploy corals out onto the reef, and really working out how we integrate into those processes. What does cryopreservation need to do to support those production and deployment processes? And you can see from some of the work we did looking at how we use cryopreserved sperm to produce larger quantities of larvae. That's sort of helping us to then understand, how many samples you need to be banking, for example, how many do you have to have in reserve, how many do you need to be using to produce larvae every year. I mean, there's a lot of industries out there that do cryopreservation at scale. The dairy industry, for example, which is a multi-billion-dollar global industry that trades in bull semen as a tool for genetic management.
Slightly more romantic version, the reef version.
Yeah. So but we can learn from those industries, right? Like those, there's been
agricultural scale approach. Yeah,
absolutely. There's been millions of dollars of investment into developing those tools and pathways, and so we can benefit by utilizing that technology and figuring out how it works best for coral, adapting it for our needs, and then integrating it into those restoration pathways.
And you mentioned as well, I guess, all of us in the reef world are very, very familiar with the restrictive reproductive process, these magical 2 nights a year when the reef, explodes into life 5 days after a full moon in November or December, depending on where you are. So there's extreme temporal limitations. So it's a little different to those agricultural processes. Can what you're doing in cryopreservation, and when we look at reef seeding or growing corals in
Mm, mm.
aquaculture to deploy, spawning is that bottleneck. Can
Yeah,
cryopreservation help us alleviate that bottleneck?
yeah, yeah,
Build out that
absolutely.
window?
To some extent. I mean, there's so cryopreservation of sperm is great. That allows us to do things like, cross individuals that are spawning in different weeks or nights or years or whatever, right? So we're not limited by which corals happen to be spawning on the night. Um, but you still, to use cryopreserved sperm, you do still need access to eggs. Unfortunately, it's currently not possible to cryopreserve coral eggs. Um, but so one of the ways that we can kind of overcome that limitation potentially is through cryopreserving coral larvae. I think that's really where the step change can come from. As I mentioned, we're kind of restricted at the moment to only having larvae available for restoration during that brief annual spawning period. But if we can bank down larvae at scale, it allows us to decouple the spawning from the deployment, to have
Yeah.
larvae available throughout the year. Yeah.
And so in terms of people in the room may not be familiar, a coral is not just a coral, obviously. There's something like 8,000 different species of corals. So if we're talking about I guess restoration in the sort of terrestrial sense that people may be
Yeah.
familiar with, when we're talking about replanting or restoring coral reefs, we're also talking about which corals we're going to restore.
Mm.
So cryopreservation, where are you guys at with that in terms of the types of corals that you're looking at?
Yeah. Yeah, I mean, there's a few challenges there in how you approach that. So historically, we've kind of tried to aim for just maximum diversity. We bank down as many species as we can, as many individuals as we can, targeting. Initially, it was 30 individuals per species was kind of a target, but the challenge with the Great Barrier Reef, places like the Great Barrier Reef, is we're still fortunate that the reef is in relatively good shape. There's still lots of populations out there, lots of healthy corals, and lots of species. And so we we can't work with everything. We can't save it all. And so we do have to prioritize and think about that. And so
Strategically, yeah.
yeah, and so as part of RRAP, we've undergone a bit of a strategic prioritization process to kind of identify a starting group of species that we want to target. And so we've got a group of 20 species, hopefully growing to 30 or more species that we want to target. And that's looking at species that exist across a range of ecological functions. So, you've got your corals that are branching, and you've got your corals that are encrusting and bouldering, and, corymbos and all these different forms and shapes that make up the structure of the reef and provide all of those different habitats. It's important that we are, sampling and working across all of those different types so that we can maintain, the future reefs that are benefiting from these restoration technologies are, good habitats and are as close to what we have now as possible.
Yeah, and I guess that's really behind the philosophy of intervention. These are interventions. So
Yeah.
how do we kind of restore a reef as much as possible? And I see a wonderful question in relation to that actually in the chat, which we'll get to. But I wanted to touch
Mm.
on I guess, well, for me, one of the most powerful aspects of this project is the integration of Indigenous leadership and cultural knowledge. And for those in the room who
Yeah.
aren't familiar, Aboriginal and Torres Strait Islander peoples have been linked with the reef for time immemorial, and there are some 70 traditional owner groups with authority for sea country management in the Great Barrier Reef Marine Park. And you've mentioned that RRAP is a program. We ensure that there's free, prior, and informed consent for all activities undertaken, and
Yeah.
touched on how we're kind of exploring beyond that baseline. And so I guess what we're really talking about is kind of setting a bit of a new standard in Australian science. So I guess I'm interested in how it's changed your perspective of this research and how you approach this research kind of locally and globally. What lessons can other
Mm.
research projects take take from this model that you're trying to, or the framework that you're trying to build collaboratively with traditional owners?
Yeah, it's been I guess in hindsight it should have been quite obvious, but it was something that kind of we weren't originally planning for it to be such a huge part of the program, but now it actually guides a lot of what we do, and that's those cross-cultural approaches to what we do. So it really has kind of changed how we think about all of this work. And what we do with cryopreservation is a little bit unique in that the samples we collect are there potentially for decades, right? So we need to be able to ensure that they're linked to country. So potentially even, in 100 years, we're all gone. There's still coral samples at Taronga that have been collected now. We need to make sure that they are still linked to country, that the people in the future who are using them are still going about getting the free prior and informed consent and all of that sort of thing. But in reef restoration more broadly, there's a lot of implications then for things like assisted gene flow, where you're moving corals between different regions to try and transfer a genetic or phenotypic advantage. So it really does kind of provide a bit of a framework for how you think about all of those kind of relocations of samples, regardless of how long they're going to be transferred for.
Absolutely. And I think it's that that thinking longer term and thinking about that traditional owner knowledge and cultural knowledge working alongside, I guess, Western science knowledge as well. That's really critical as we seek to kind of, I guess, translate these solutions into tools for the community, both here in Australia and across the globe. And I guess that sort of final point in discussion, because John, I'm aware we're short on time, but I think let's zoom out from the Great Barrier Reef, much like you did, in terms of this international perspective and this global cryopreservation efforts. RRAP has some other examples where we've used some of the solutions being developed in other areas of the program around coral seeding that have been had uptake by community here in Australia, but also internationally in the Maldives and the Pacific. So can you expand a little bit on kind of the democratic approach that we'll see in some of those training programs? It's about accessibility, but it's also about cost and modularity of solutions as well, right? Not everyone has a huge research lab at their disposal.
Yeah, that's right. That's a really important thing, because there's, while there are big programs like RRAP that have access to a lot of different technologies, a lot of the people who will actually be impacted by the loss of coral reefs are people in developing nations and places that don't have access to these technologies, where it's challenging. And so it's really important that what we're developing can be used across that range of kind of scales and economic needs. And so we are developing tools like that 3D printed freezing system, for example, that can be used at low cost. They can be used on the beach, for example, and working with some of the groups internationally on developing those things, but then also having those high-tech systems that can support aquaculture programs as well, because it's really important that if you're in groups in say the Pacific or Southeast Asia who really do rely on the reefs for a lot of their income or food, that they have access to these technologies, and there are versions of these technologies that are actually suitable for use in those situations.
Yeah, absolutely. And I think if people aren't aware, all of this work is the Reef Restoration and Adaptation program is a public good program. So the idea is that we have the benefit of all this wonderful research support here in Australia, and we can start to share that knowledge, and those solutions internationally. So I think that's absolutely critical. John, it's a short discussion, but I'm conscious that we're slightly over time for your allocated schedule. John Tasker, if you're there. So I might
Sorry, I ran a little long.
No, not at all. Yeah, so there's a really great question here. Oh,
we do have time if anyone else wants to ask any questions. So please, we've got another 10 minutes or so.
I think one of the questions that I had that I jotted down here is going back to the discussion just a bit around the cultural safety and protocols. One of the reflections that I've had through working in this program is obviously there's as much cultural difference across the reef as there is anywhere else with traditional owner groups. So you've set the standard on Konomi Island and with the Wapabara traditional owners, but in terms of sort of expanding out into other areas, what does that process look like in terms of the flexibility of that framework?
Yeah, absolutely. That the work we've done with the Wapabara is a starting point. We've collected from across 10 different sea countries over the history of the program, and so that framework allows us to, I guess, have that discussion as a starting point with those other groups. And so we are working with the AIMS Indigenous Partnerships team, for example, on thinking about how we would do another series of workshops with those other traditional owner groups to talk to them about what it means to them, because we can't assume that all of those groups will take the same views or have the same concerns or views as the Wapabara folks did. So having those discussions will be a really important part. But then this work that we've done to date, it provides that framework, that kind of a starting point for discussion. So we can say, here's what we've got so far. Here's what we think might work. What do you think? What else could we do? Or what's relevant to you? And so there's a lot of traditional custodian groups, but at the same time, it's not a difficult thing to imagine that we would have tailor-made for each of those groups, kind of a thing. It's the right way to move feasible forward, and it's the way to do it. And so we're going to make every effort we can to do that.
And just really quickly before I get to this question in the chat, I think there was another question that I had while we were chatting as well. Just that sort of lends me to, I guess, taking everyone behind the scenes a little bit in terms of the biorepository management. Not everyone gets to visit Taronga Zoo and see, the glass door and see behind what some of the facilities look like. But what we're really talking about is a legacy piece here. So we're not talking about biorepositories now. We're talking about being able to access and source them 10 years, 20 years, 30 years into the future. So you're actually managing long-term, not just short-term, right?
Yeah, absolutely. And, this is one of the challenges with biorepositories or biobanks all over the world. I mean, Taronga is a state government agency, which means that there is that, that kind of longevity to the program. And that's the same with the Smithsonian in the United States, for example. But, and so part of that work that we were doing is with CORDAP in developing those regional nodes in Curacao and in Mexico was about having a similar kind of stability for biorepositories in other locations, because we it's very easy to collect samples and just put them in a tank under a bench, and then the money runs out or the researcher retires and the samples are left behind. So it's really important that these biorepositories are ideally linked to government funding of some sort to have that kind of longevity, but also those database systems are essential because you have to know what you've got and where it came from. So just having that kind of infrastructure and process around the biobanking work is really essential to have functioning biorepositories that are going to be that resource for the future.
Yeah, and that will be the skeleton that will or the foundation of then supporting a democratic reef restoration industry that is
Yes.
delivered by reef communities. Yeah.
Yeah, absolutely. Because not every country is going to have the capacity to have a huge government-funded biorepository. But if there are these regional nodes and that can provide that stability, and then if you can provide those transferable training tools that can go out to all of those communities, then you create a system where biobanking can be taking place in reefs around the world, regardless of means. And that's a really important thing as we sort of think about securing as much of the genetic diversity as we can while we can. Yeah.
Absolutely. And I think, John, if you're comfortable, I'll start off,
Yeah.
I guess, Odette's question here, and then, jump in with cryopreservation considerations. But thank you, Odette, for this question about the assisted evolution, which really for us within RRAP is nothing to do with genetic manipulation. It's about enhancing a natural process, so crossbreeding, hybridization. And ethical considerations around putting or replanting those corals back onto the reef. So I think what's really critical is we're operating in the Great Barrier Reef Marine Park, which is a very closely managed and highly regulated system, and we operate under research permits. So we're an R&D program. And so we are doing a lot of this work in the lab, but we are completely subject to the Marine Park Authority and their management decisions as to how we then translocate, if we even translocate those corals back into the wild. This is just the governance perspective. John can talk biological perspective. Pardon me. So yeah, we're really subject to that, and that's an ongoing process, through this R&D journey, and also as we start to pilot really small scale trials, we do it at small scale and then slightly larger scale, and so on, so on, so on. So things happen slowly and with feedback to the marine park and its management system as well, because we're really kind of uncovering what are the considerations, what are the risk considerations. There's actually, it's kind of like building the train at the same time it's moving at 10, 100 kilometers an hour. We have to unlock the fundamental questions and answers, as well as the applied science questions and answers, as well as how do we even help corals become more heat evolved, but what is the fundamental impact on ecosystem? So with our program structure, we cross-cut with ecological intelligence teams who can answer some of those fundamental questions of those early recovery and what assisted evolution or more heat-tolerant corals are doing out on the reef naturally. We also have risk management groups who independently look at what the different impacts of these interventions are. And actually our chief focus, and if you jump onto our website, it should be published in the next few weeks, one of their chief focus has been on what are the ecological, social, economical, etc. risks and considerations around putting heat evolved corals out onto the reef. But John, you may want to speak to that more broadly or more biologically, I guess.
Well, I mean, just to say that these are challenges that are being faced by managing agencies all over the world, right? So Florida is very similar, or it's all heavily managed in terms of what can go on and off the reef, for example. And traditionally, marine park managing agencies, it's always that do no harm approach, and so they really need to be quite certain about something before it will be allowed to go ahead. So that's a strong way to mitigate. That's right. And so that's a good way to mitigate the risk. But then also that's balanced with how reefs evolve into the future and what risk actually means in terms of is it more of a risk to do nothing versus the risk of doing something. And so that's that balance that managing agencies are facing all over the world at the moment.
Yeah, absolutely. And look, John, John Tasker, we've just hit time. I can see. I'm just going to pop our website as well in here. If anyone wants sort of to follow along with RRAP information and journeys. But I'll throw back over to John Tasker.
Thank you so much, Jane, John. That has been a fantastic conversation. We've learned a hell of a lot. So we might leave it there. Thank you so much. And I'm sure we'll speak again for those online again soon.
And John, please let anyone know, if there are any follow-up questions.
Yep.
Send through an email to myself. And John, John Daly, I'm volunteering
please
you
get
as
in
well.
touch. Yep. Yeah, definitely.
Perfect.
Yeah, please reach out if you got any questions.
Yes, well, thank you again. Brilliant. And thank you all those for joining us. We'll speak again soon. Bye.
Awesome. Thank you. Bye.